Hi, welcome to our fifth lecture, Palladium-2-Catalyzed Stereoselective Formation of alpha-O-glycosides. Carbohydrates are the most highly diverse class of biomolecules in nature. It is well known that carbohydrates play a vast role in biological processes. Some of these roles include cell-cell recognition, cellular transport and metabolism, storage and transport of energy, functioning of the immune system, fertilization, pathogenesis, blood clotting, and adhesion. The addition of saccharides to proteins or lipids, known as glycosylation, is a major post-translational modification of membranes and secreted proteins in cells. Alternations in the structure of glycans and polysaccharides are associated with a variety of diseases, including metastatic cancer. Investigation into carbohydrates has increased over recent years due to their importance as building blocks, biological tools, potential drug candidates, and synthetic targets. For example, Boons and co-workers have successfully synthesized the first three-component synthetic carbohydrate anti-cancer vaccine, and have demonstrated that it elicits an immune response through immunoglobulin G antibodies. Manopeptymycins, newly discovered class of glycopeptide antibiotics produced by Streptomyces hydroscopus, LLAC98, have shown antibacterial and mechanistic activities. Therefore, a glycosylation methodology to stereoselectively construct the cyclic glycopeptides is of synthetic interest. Here we show manopeptymycin E. This inhibits methicillin-resistant staphylococci and vancomycin-resistant enterococci. A cyclic hexapeptide core with alternating D and L amino acids is present. Also, an N-glycosylated beta-hydroxyuracil and an O-glycosylated tyrosine. Those are the synthetic structures of interest. A variety of glycosylation methods have been developed for complex carbohydrate synthesis due to the critical role carbohydrates play in metabolic pathways. In forming glycosidic bonds, the glycosyl donor and the glycosyl acceptor are the two required components. A glycosyl donor is referred to as the carbohydrate unit that donates its anomeric center to the glycosidic bond, and the glycosyl donor is referred to as the unit that receives the anomeric center. In general, a promoter or catalyst is needed to activate the electrophilic glycosyl donor anomeric leaving group, and then is coupled with the appropriate nucleophilic glycosyl acceptor to generate a glycoside as a mixture of alpha and beta anomers. Here we see upon activation an intermediate where the glycosyl acceptor can approach either the alpha or beta face. Utilizing an ester in the equatorial position to block the alpha position affords exclusively the beta glycoside. Utilizing an ester in the axial position, however, affords the alpha glycoside. Many challenges exist with utilizing the antichimeric assistance. For example, in addition to forming the desired beta-glycoside, the glycosyl acceptor can attack the acetyl carbon, or hydrolysis of the acetyl could occur. In 1901, Koenigs and Knorr reported the treatment of 2,3,4,6-tetraoacetyl alpha-D-glucopyroxenol bromide with silver carbonate in the presence of methanol to provide beta-DO-methylglycoside. The stereochemical outcome of this reaction is influenced by the presence of neighboring group at the C2 position that via antichimeric assistance provides the formation of a 1,2 trans stereochemical rearrangement. This general ester provides a good neighboring group participation, whereas ethers do not, leading to a mixture of stereoisomers. Several disadvantages exist in Koenigs-Knorr glycosylations. First, the unstable glycosyl halides are prepared using harsh reaction conditions. In addition, The toxic and sometimes explosive coactivators are generally used in equal molar quantities. Furthermore, glycosyl halides are prone to undergo 1,2 eliminations, or hydrolysis. Lemieux and co-workers introduced a mild glycosylation method that limited the use of stoichiometric amounts of heavy metals. Lemieux's method utilized glycosyl bromide or chloride in the presence of a phase transfer catalyst, such as tetrabutylammonium bromide, to yield the corresponding glycoside. This methodology led to the elegant synthesis of several blood group antigenic determinants. In 1981, Mukiyama and co-workers introduced a method closely related to the Koenigsegg glycosylation, where a considerably more chemically stable glycosyl fluoride donor is utilized in place of its glycosyl bromide and chloride counterparts. The major advantage of this methodology is the ease of preparation of the glycosyl fluoride donors. These donors are prepared under mild conditions from the corresponding hemine acetal via the use of diethylaminosulfur trifluoride, or DAST. Due to the synthetic flexibility of glycosyl fluorides, a diversity of fluorophilic activating reagents have been found to promote glycosylation, including silicon tetrafluoride, TMS triflate, and boron trifluoroetherate. Kahn and co-workers have reported the use of sulfoxides as effective latent leaving groups. In the reaction, glycosyl sulfoxides were activated with trifluoromethylsulfonic anhydride and toluene at -78 degrees Celsius, and subsequently coupled with a glycosyl acceptor to yield glycoside. The Kahn method proved to be efficient in the preparation of glycosides from sterically hindered or otherwise unreactive substrates. Unfortunately, the stereochemical outcome of the coupling is dependent on the glycosyl donor and glycosyl acceptor protecting groups. This method goes through a glycosyl triflate generated upon reacting the sulfoxide with triflate anhydride at -78 degrees in toluene. The use of glycol derivatives as glycosyl donors has recently been used in pi-allyl palladium strategies for the stereoselective synthesis of O-glycosides, as shown with O'Doherty's work. However, as seen with previous glycosylation methodologies, O'Doherty's strategy is substrate dependent, thus relies heavily on protecting group manipulation. O'Doherty's strategy is dependent on a de novo pi-allium-based glycosylation that utilizes a highly reactive pyranones as glycosyl donors. Some advantages to this palladium-based strategy are the glycosylation proceeds with high diastereoselectivity. The palladium is used in catalytic amounts, and the reaction conditions are mild in the sense that they use stoichiometric amounts of Lewis acid promoter is avoided. However, due to poor reactivity of the glycosyl donors as well as the alcohol nucleophiles, this pi-allyl palladium glycosylation strategy is limited to the relatively more active pyranone donors. Lee and co-workers were able to increase the reactivity by utilizing the zinc 2 ion to activate both the alcohol acceptors for the nucleophilic addition and the glycol donors for ionization. A major disadvantage to Lee's approach is lengthened reaction times. Currently, many efforts are being concentrated on developing new methodology and reagents for the construction of isolated glycosyl donors, which react with nucleophilic glycosyl acceptors to form glycosidic bonds. In all these methods, the stereoselective control of the glycosidic bond is dependent on the nature of the substrate. Often, very burdensome and tedious protecting group manipulations on the substrate are required in glycosylation reactions to control the anomeric selectivity, resulting in the addition of multisynthetic steps and decreased yields. Therefore, the development of a methodology that does not rely on protecting groups for anomeric selectivity is needed. Here, I will present my efforts to develop such a methodology that utilizes mild palladium-catalyzed reaction conditions. A novel method for the stereoselective construction of alpha-O-glycosides has been developed utilizing glycols as glycosyl donors. The alpha-O-glycosides can be directly derived from glycols through the use of palladium-2 ligand catalyst. which is further described in this lecture. In this reaction, it is believed the glycol pi system is activated by the palladium-2 ligand complex for the stereoselective attack by several external oxygen nucleophiles. The trichloroacid imidate group functions as the leaving group as well as the directing group for the palladium-2 catalyst to the double bond of the glycol. One major advantage to this strategy is the anomeric control relies on palladium ligand catalyst donor complexation instead of the nature of the protecting groups of substrates. This eliminates tedious protecting group manipulations that are often associated with glycosylations. Obtaining alpha-glycosides is attractive as it opens up the pathway for a broad scope of alcohols and diols. It is important to note that although the 3,3 sigmatropic rearrangement of allylic trichloroacidimidates was pioneered by Overman, his method was never employed in carbohydrate synthesis to control the anomeric selectivity. Initial studies focused on optimizing reaction conditions utilizing the Glucal intermediate product here, which was prepared in 2 steps from D-Glucal. D-Glucal was treated with 2,2-dimethoxypropane and DDQ and acetone to yield the 1-hydroxyl sugar. Subsequently, the thermal and chemically stable trichloroacidimidate glycosyl donor was synthesized from the corresponding 1-hydroxyl sugar by treatment with trichloroacetonitrile in the presence of DBU. With a method in hand, a variety of imidates were prepared. And here's some more imidates that we prepared. With access to the glycosyl imidates, efforts then focused on the feasibility of a palladium-2 catalyzed stereoselective formation of alpha-O-glycosides. Initial treatment of glucal imidate with 5 mol% of palladium bis-benzonitrile palladium dichloride, 2 equivalents of paramethoxyphenyl, and in DCM at 25 degrees for 35 minutes, provided the glycoside in 45% yield with alpha-beta selectivity 2:1, along with there was a significant amount of decomposition. Decreasing the amount of palladium loading to 2.5 moles still afforded the product, but this time in 46% yield with alpha-beta selectivity 4:1. Changing the solvent from dichloromethane to toluene slowed down the reaction. The desired product was obtained in 43% yield with going back to an alpha-beta selectivity of 2:1, along with a significant amount of undesired product observed. The next aryl alcohol nucleophile examined was the sterically hindered 1-naphthenol. Under optimized conditions for paramethoxyphenyl, glycoside was obtained in 55% yield with alpha-beta selectivity of 3:1. Adding triphenylphosphine increased the yield to 66%. It was hypothesized that the anomeric selectivity could be dependent on the palladium ligand. Accordingly, an assortment of Buchwald sterically bulky biaryl phosphine ligands were screened. Treatment of the glucal intermediate was performed with a preformed mixture of bis-acetonitrile palladium dichloride with John Fau's ligand in a 1:1 ratio. Had no effect on the outcome of the reaction. It was likely that the palladium-phosphine complex may have not been in the reaction, since it is known that the rate of ligand exchange for bis-acetonitrile palladium dichloride is slow. A significant improvement occurred when bis-benzonitrile palladium dichloride was used in place of bis-acetonitrile palladium dichloride. Furthermore, anomeric selectivity slightly increased from alpha-beta 3:1 to alpha-beta 4:1. After screening 7 of Buchwald's bulky biaryl phosphine ligands with our new palladium catalyst, it was found that DTTBP provided glycoside in good yield with excellent selectivity. Here, to our excitement, we saw an 84% yield with 20:1 alpha-beta selectivity. All reactions were carried out with a concentration of 0.2 molar, 2 equivalents of 1-naphthenol, 2.5 mol% of phosphine, and 2.5 moles of palladium-2 catalyst in dichloromethane. These results suggested a more bulky biaryl ligand increases the yield and anomeric selectivity of the reaction. A proposed mechanism for the palladium-2 catalyzed stereoselective formation of O-glycosides is shown here. Ligand exchange between benzonitrile palladium dichloride and DTTBP occurs to yield the palladium phosphine complex. This complex then undergoes reversible coordination to both imidate nitrogen and alkene of the glucal to form the palladium-alkene complex. Subsequent migratory insertion will provide the oxonium-palladium sigma complex. Nucleophilic attack by the glycosyl acceptor on the beta phase is sterically blocked by the bulky biaryl phosphine ligand. As a result, 1-naphthyl approaches the alpha phase, leading to the glycoside intermediate. Deoxypallination and subsequent dissociation then yields the alpha-glycoside. In this catalytic cycle, 1-naphthyl acts as both the nucleophile and proton donor. In order to make sure this reaction does not go in pi-allyl palladium mechanism, a control experiment was performed. The major concern was that the electron-rich phosphine would reduce palladium-2 to palladium-0. The glucal intermediate was treated with preformed solution of 2.5 mol palladium DBA and DTTBP in the presence of 2 equivalents of naphthyl to yield glycoside in 26% yield with an alpha-beta selectivity of 1.5 to 1. The major product was the 3,3 sigmatropic rearranged product, which was isolated in 41% yield. Had the mechanism gone through a pi-allyl palladium intermediate, there would have been no control of stereochemistry, and the racemic mixture of alpha-beta atomers would have been observed. Our primary concern was the electron-rich phosphine was reducing our palladium source from palladium-2 to palladium-0. However, our results showed that clearly our mechanism did not proceed through a pi-allyl palladium mechanism. To test the feasibility of this novel palladium-catalyzed stereoselective methodology in terms of O-aryl glycoside synthesis, several glucal donors with a variety of protecting groups were synthesized. As you can see, we achieved mostly alpha selectivity, some were exclusively alpha, and the yields were moderate to good. We then turned our attention to a range of Glucal donors and observed exclusively alpha-O-aryl glycosides with good to excellent yield. Extending this methodology to form O-glycosides from aliphatic alcohols was then investigated. Due to the poor nucleophilicity of benzyl alcohol, it was initially converted to the relatively reactive potassium alkoxide. The glycoside was obtained in 21% yield exclusively as the alpha atomer, along with the recovery of galactyl imidate in 53% yield. Next, benzyl alcohol was converted to the relatively more reactive zinc 2 alkoxide. Accordingly, treatment of the glycol intermediate with a preformed solution of benzyl alcohol and zinc diethyl zinc and 2.5 moles of palladium catalyst in DTTBP with a mixture of toluene and DCM 1 to 2 provided the glycoside in 68% yield, along with the rearrangement product in 22% yield. In this reaction, 2,6-ditert-butylphenyl was used as a proton donor in order for the palladium phosphine catalyst to turn over. Decreasing the amount of benzyl alcohol significantly increased the yield of the glycoside to 77%. The undesired product obtained with toluene as the solvent is believed to be the alpha-glycosyl-mid, which is formed via the allylic imidate rearrangement. In order to minimize this occurrence of this rearrangement, dichloromethane was used as a reaction solvent for the rest of the glycosylation reactions. Having shown the current palladium-2 method was feasible with benzyl alcohols as the glycosyl donor, this chemistry was further explored with a variety of aliphatic alcohols. Treatment of the glycol intermediate with a preformed solution of zinc 2-alkoxide proved successful and provided the desired glycosides, most of which were exclusively the alpha anomers. Several aliphatic carbohydrates were examined, including furan, ramenose, and glucose-based nucleophiles. Several products were then oxidized by Dr. Mercer into the synthetically desirable cis and trans diols. Using glycols as starting material, a novel method for the stereoselective construction of alpha-O-glycosides has been developed. The scope of this methodology can be applied to both aryl and aliphatic alcohols as glycosyl acceptors. For the reaction to proceed with aliphatic alcohols, the glycosyl acceptor needed to be softened by converting them to the corresponding zinc alkoxides. The dry chloroacetate imidate of the glycosyl donor serves as both the leaving group and directs the coordination of the palladium-phosphine complex to the Glucal pi system. As a result, the palladium-phosphine complex sterically blocks the beta phase of the glycosyl donor from nucleophilic attack, resulting predominantly in the alpha anomer. In the case of galactols, strictly alpha anomer is observed due to the acyl hydroxyl at the C4 position. This approach proceeds via mild glycosylation conditions with shortened reaction times at room temperature. In addition, anomeric selectivity is reagent-based as opposed to traditional substrate-based glycosylations, where tedious protecting group manipulations are often employed. So here I presented to you a novel method for the stereoselective synthesis of alpha-O-glycosides. I would like to acknowledge Professor Glenn. He was a huge support in this research. Professors Paul Grieco, Dave Single, and the late Professor Dr. Alt. And Scott and Rachel for help with the structure elucidation of these compounds. And the other members of the Gwen group, especially Jae Moon Yang. He was a huge help, along with Greg Mercer. Thank you.